Hello and welcome to episode number 21 of Gowing Life's Longevity Weekly with me, Chris Cohen. This is where I bring you all the news and updates from the world of longevity, and today we're going to be talking about what is telomere attrition, my chat with Professor Michael and Professor Irina Conboy, and also part 3 of the article series that I'm writing on studying neurological disease with induced pluripotent stem cells. So without further ado, let's get into it. So firstly, what is telomere attrition? Our bodies experience over 10,000 trillion cell divisions in its lifetime. Successful cell division is absolutely crucial for the repair and growth needed to maintain life. The most important part of each single division is the accurate replication of the cell's DNA. Now, each time a cell divides, it must make a near perfect copy of its genetic information to pass on to its daughter cells. However, the molecular machinery that carries out this copying process cannot quite replicate the entire length of DNA, meaning that a portion of the DNA at the end of each chromosome is lost each time the cell divides. So where do telomeres fit into the equation? Well, telomeres are sequences of repetitive DNA that serve to protectively cap the ends of the chromosomes. However, unlike the rest of the genetic code, telomeric regions contain no actual useful genetic information and can be likened to a plastic tips on shoelaces, protecting the ends of the chromosomes from damage. Now, when a cell divides and its DNA is copied, a section of the telomeric DNA is lost. This is called telomere attrition or shortening. Thus, telomeres allow cells to divide without losing crucial genetic information. They also act as markers, preventing chromosome fusion from occurring, a process which sees the ends of different chromosomes fuse together, dramatically altering the cell's genetic makeup in a way that can lead to malfunction, cancer, or even cell death. So how does telomere attrition relate to aging? Well, telomeres are only able to protect the chromosomes for so long. Every time the cell divides and the DNA replicates, the telomeres will become shorter and shorter. In some cells, an enzyme called telomerase is able to repair these telomeres, but the majority of normal mammalian cells do not express telomerase, meaning that telomeres will continue to shorten. To prevent this from happening, cells will usually stop dividing when their telomeres become too short. This is called replicative senescence, or they'll commit suicide in a process called apoptosis. This is called the Hayflick limit, and occurs in human cells in lab conditions after about 40 to 60 divisions. Now, telomere exhaustion is observed in aging, and reduces the ability of tissues to repair and regenerate themselves. Meta-analyses indicate that shortened telomeres are correlated with increased mortality risk in humans, suggesting that telomere length may be an important factor in lifespan, though questions remain about just how important it is. Mice genetically modified to have longer or shorter telomeres have longer or shorter lives, respectively. In both humans and animals, reduced expression of the telomere repair enzyme telomerase correlates with increased disease incidence, while genetically enhancing the expression of telomerase makes mice live longer. This hints at the rate at which telomeres are degraded or repaired may be a more important factor than their initial length when it comes to an organism's lifespan. So to find out more about these studies and delve a little bit deeper into telomere biology, go and check out Alex's article. And secondly, a few months ago we were absolutely privileged to host the husband and wife team, Professor Michael and Professor Irina Conboy, to chat about their research on the blood's role in aging and their fascinating paper titled Diluting Blood Plasma Rejuvenates Tissue, Reverses Aging in Mice. So here's a little clip from it. So could you tell me the results of your recent study?
Sure. So let me do a quick bridge between the parabiosis, right? So the parabiosis is like a Siamese twinning of the young and the old animal. And they share blood, but they also, because they're connected for a very long time, a relatively long time, they share the tissue and organ systems of the partner. So the young animal suffers half its mass or more than half its mass being old and having old liver, old kidneys, blah, blah, blah. The old partner gets access to a young set of liver and kidneys and pancreas and insulin regulation and digestive system and whatever. So we didn't know for sure if it was the effects that we saw were because of the blood or because of the other systems that kind of communicate through the blood. The next experiment we did was a blood exchange. So we just took two animals and transfuse blood back and forth between them. And then after several exchanges, you approach homogeneity between them. So there's about 50-50, and then disconnect them. And because the only thing that was transferred was the blood, we could rule out the effects of organs and regulatory systems. So from that work, we found that the liver was improved in the old animal. The muscle regeneration was improved. The brain wasn't really all that improved. And the young partner that got old blood, the muscle regeneration was a little worse. The liver couldn't really see much of a defect for the things that we looked at, but their brains looked like they got stupider.
So thanks again to both Michael and Irina, two giants in the field, for such an enlightening conversation, and you can check the full video out in the description below. And finally, a couple of weeks ago, we looked at part 2 of an article series looking at studying neurological diseases with induced pluripotent stem cells, or iPSCs for short. Now, we looked at how we derive these handy cells and why they're so useful when it comes to studying disease. However, they do have their limitations. The main flaw of iPSCs when it comes to studying neurological disease is their two-dimensional structure when grown on a cell culture plate, which is obviously vastly different from the 3D structure of an actual human brain. Now, this failure to replicate the 3 dimensions of a real-life brain reduces the model's capacity to accurately mimic real life. Recent innovations in biomaterials and stem cell cultures have allowed the generation of complex 3-dimensional models known as brain organoids, which showcase a significant step forward with respect to 2D models, allowing the formation of tissue architecture and complex neural networks far more akin to that of a real brain. Now, to get from having a population of induced pluripotent stem cells on a plate to a brain organoid requires roughly 2 months of directional differentiation of these stem cells into brain cells. This is done via the use of specific proteins known as patterning factors, which were found to drive the process of brain development in the growing embryo. Now, as the organoids develop and grow, it forms the grooves and troughs characteristic of a human brain. These can then be maintained for up to a year. Now, full brain organoids mimic the entire human brain, whereas regionally specific organoids are designed to replicate different areas. Combining one or more of these regionally specific organoids can help to model the interactions between 2 distinct regions in detail, which would not be possible for a full brain organoid. Now, there is a continued strive to improve these 3D models, one aspect of which is to achieve organoid vascularization. An established system of blood vessels would enable the organoid to sustain growth over a longer period of time, and to improve the accuracy of the in vivo environment it attempts to recapitulate. Now, another aspect is to introduce an immune system to the organoid. The immune system of the brain differs from the rest of the body, as it is composed of cells called microglia. Microglia are a specialized population of macrophages, found in the central nervous system, which boast a range of functions that help to maintain the cerebral environment in its optimal conditions. Now, in conclusion, the development and continued improvement of these 3D brain organoids has provided an environment that more closely resembles that of a real brain. This provides a more intricate and elaborate neural network in which to recreate and model these neurological diseases. Now, keep your eye out for part 4, where we will be looking at real-life cases in which the use of these brain organoids has facilitated some groundbreaking discoveries in regards to age-related neurological conditions. And that's it for this week's episode of Gowing Life's Longevity Weekly, so I really hope you've enjoyed it. If you'd like to go and check out more, then go to the Gowing Life website, gowinglife.co.uk, where you can check out all the articles we've been writing this week, including Alex's Hallmarks of Aging series, and how coffee may protect against liver disease. So go and check it out. I hope you've enjoyed it. Have a great week, and I'll see you again next time. Thank you very much. Goodbye.